So I am Nick Courtwright. My paper was also on polyether ether ketone. So the title of my paper was Effects of Bioactive Glass and Beta Tricalcium Phosphate Containing Three-Dimensional Laser-Centered Peak Composites on Osteoblast in Vitro. A little vocabulary. Osteoblast are cells that synthesize bone, and in vitro just means in glass outside of the body. So this paper was from the Journal of Biomedical Materials Research. The specific scope of this paper was to evaluate laser-centered PEEK for use as a bone implant material. PEEK is already used as a bone implant material, however it's normally made by an injection molding process, and there's a need for a more direct manufacturing process to create complex geometry. So this is where laser sintering comes in. The key question addressed in this paper is, what are the effects of laser-centered PEEK with incorporated bioactive bone materials, specifically bioglass and beta tricalcium phosphate, on osteoblast in vitro? The materials used in this experiment are PEEK along with carbon black, beta tricalcium phosphate, and bioglass. The carbon black was added to increase the flowability for the lasering sintering process. The beta tricalcium phosphate and bioglass are what's known as bioactive materials. And with an implant, you often have fibrous tissue that grows and isolates the implant from bone. And these bioactive materials help prevent that. So you see here the one on the left is just pure PEEK, A here. B is PEEK mixed with carbon black. C is PEEK, carbon black, and beta tricalcium phosphate. And D is PEEK, carbon black, and bioglass. So the process conditions examined in this experiment were cell morphology, cell viability, and cell proliferation. And these were the tools that were used to measure these are the scanning electron microscope and a spectrophotometer. The first result from the study is, how does incorporating bioactive materials into laser-centered PEEK affect cell viability? So this is a picture on the left of actually looking down and how they measure the cell viability. And the table on the right is comparing each of the samples and their mean values of cell viability. So as you can see, PEEK Pure has a mean value of 95.3% cell viability, and adding the 1% weight carbon decreased it slightly to 93.2%. With adding beta tricalcium phosphate decreased the viability to 84%, while bioglass increased to 95.5%. The second key result from the study was how incorporating the bioactive materials into the laser-centered PEEK affected cell proliferation. And this chart shows measuring the cell proliferation at days 3, 7, and 14. From this chart, it can be seen that carbon black had no statistical effect on cell proliferation, while beta tricalcium phosphate resulted in a lower proliferation at each time, and bioglass had the highest proliferation. So the summary for this paper is cytotoxicity, which just means toxic to cells, is not induced on any laser-centered PEEK examples, and this is proven by the viability and proliferation test. Beta tricalcium phosphate from this study showed that it had no favorable effects on proliferation, while bioglass shows advantageous effects. So from this paper, it helps prove that laser-centered PEEK parts are good candidates for bone implants, and it also introduces the possibility of adding these bioactive materials to the PEEK for increased bone binding.